Welcome to the second last day of April. Does anybody feel like April has had 8,000 days in it? Oh my gosh. April has just, but we've been in quarantine or whatever you want to call it all this time. So I guess that's why, but holy cow. Okay. So just a reminder that the test will open on Friday, maybe Saturday. I don't know, depending on when I get it done. And then it'll be open through Monday, the 4th of May. And then,
Like Friday morning or Friday after class?
I don't know.
Okay.
You're not going to take it then anyway.
I kind of want to take it Friday.
Well, it'll be open sometime for you on Friday. I don't know when, but it depends on what I do tomorrow.
But no, I'm not going to wake up at midnight to take a test.
No, I know you're not. I don't know. It depends on, because I got to clean up the lab today. They want to mop or wax my floor in there. And so there's still models on the table. I haven't done anything. We just left and I left it. So anyway, so I'm recording this. I better get on with what we're doing. So it'll be open sometime on Friday. All right, so let's go ahead and get into where we left last time. So let's just review the different pre-embryonic stages. So fertilization, oops. Come on. Okay, so fertilization results in a zygote, and then the zygote will start going through mitosis. Sorry about that. Oops. And mitotically divide, and remember that the blastomeres, that's what these little cells are called. They don't increase in size, so they stay small. They don't grow after replication, so they just divide and divide and divide and divide until you get to the morula. And so there's the morula right there, the solid ball of cells. And then you start to get a cavity on the inside, the blastocyst cavity. And so that will result in a trophoblast on the outside, so the chocolate part of a wonder ball. And then the inner cell mass, of course, will then be what develops into the embryo. And so at this point, it's 6 days. We will have implantation occur, and then we'll proceed on through the rest of the pregnancy, barring any millions of things that could actually happen to cause that pregnancy to not be successful. All right, so what's going to happen in implantation? So 6 to 7 days after fertilization, we're at day 21 in the female. So she's not going to be having a period yet. So, you know, if we have a 21-day cycle, everything's like shifted back. So we're still fine. We're still halfway through the luteal phase. Let's put it that way. And then we're always sure that we're right. We're halfway through the luteal phase. No risk at the moment of miscarriage, of losing or starting a period. So we're going to do a couple things. So the trophoblast that's over the top, so we have to orient. How are we going to implant? So I'm going to go back to this picture. You know, we could implant anywhere along this circle, right? But the part where we're actually going to implant is the part of the trophoblast that's up here above the inner cell mass. So you can see in this picture right here, there's the little pre-embryonic inner cell mass that's touching up against the endometrium. If we go to this picture, we see the same thing. Here is the trophoblast that's over the top of the inner cell mass, and that's what's going to go into all of this right here. This is what's going to form the placenta and the amnion and the baby and the yolk sac. So actually the yolk sac comes from inside of here. So this is what's going to develop into the placenta, is the trophoblast that's over the inner cell mass. And that's what it says, something like that. Trophoblast over the inner cell mass adheres to the endometrium, secretes enzymes and growth factors against the endometrium, basically eliciting an inflammatory response. okay, like a mosquito bite or something like that, where we're going to, which is what the baby wants. We want to cause inflammation to occur so that we get histamine, prostaglandins, and other things that are going to cause the blood vessels there in the functionalis layer to become more permeable and to vasodilate, so that now we are getting nutrients to the implanting blastocyst there within the endometrium or within the functionalis layer. Now the trophoblast is going to form two layers once, and I'll show you that in just a sec. Once it butts up against the endometrium, then it's going to divide so that one layer is going to become the placental membranes, and then the layer of underneath is going to become the chorion, which we saw in lab is going to develop into the blood vessels on the fetal side of the placenta. The blastocyst will then be covered over and sealed off. This takes about a week. So now, once the blastocyst is sealed off, we're at day 28. And so it's embedded itself within the endometrium. And now, the most important thing of all, that embryo, those trophoblast cells, must now start to produce human chorionic gonadotropin. So human chorionic gonadotropin, oh, let's look at the name. Chorionic, so it's coming from the chorion, gonadotropin. So what does a gonadotropin do? Well, it's going to act as LH. Okay, so it's a tropic hormone that's going to go to the corpus luteum, and it's going to act as if it were LH, and it's going to tell the corpus luteum to keep producing progesterone. So it's going to produce progesterone so that we can maintain that uterine lining for the first 3 months of pregnancy. And then after that, the placenta is well enough established that it will do everything itself after that. It doesn't rely on the mom anymore. It's like it's become this independent organism. Well, it is, said new baby. But the placenta kind of is like it's an independent organ feeding the baby, but yet drawing its source. Oh, that sounds like a horror movie. Drawing its source of nutrients from the mom. Oh my heavens. Yeah, you could seriously write a really super good scary movie just with what already happens in your body when you get pregnant. I love it. Okay, so HCG is that hormone that is excreted in the urine that we test when we're doing a pregnancy test. So all of those, you know, pee on a stick and watch for the blue lines to show up or whatever. That's what we're measuring. So we have chemicals that react to the presence of HCG on that test strip, and so when they bind and they're detected, then the blue line shows up. We'll talk more about that in just a minute. All right, so I want to show you again. So we took a look at this picture up here, and we saw here's the trophoblast above the inner cell mass, button up against the endometrium. Now it's gonna invade the endometrium. It's gonna send out these like projections that will form the fetal membranes or the placental membranes. Now, if you look at here, we had cells, the cells divided, and so we actually should have layers like, you know, stratified squamous epithelium. However, the cell membranes are going to disappear between the cells, and we're going to make these big cellular masses, syncytia. And so there's one, and that's what helps it invade and just, you know, permeate through. Permeate's not the best word, but you know what I mean, like fungal rhizomes and things like that are just, ooh, they just work their way through. So that is this right here called the syncytiotrophoblast, is that the cells that were up here, as they divided, they all grew together. And so that's why there are these different circles, because those are the nuclei. So there would be 3 cells that were here together. So this little mass of cells right here, these membranes are going to disappear. And then it's just going to be part of that big mass. And so here were 2 cells. Here were 4 cells that came together. And I don't know what's going on with that weird thing. Okay, the second layer of the trophoblast, just this portion right here, is going to be the cytotrophoblast, which will give rise to the chorion. So all of this, let me draw that. Let me show you what I'm talking about when I say all of this. Let's do blue. Okay, so all of this right here to there, okay, all of that is going to become placenta for the baby. And then this right here is going to be the baby, the amnion, and the yolk sac from this inner cell mass. Now here's what it looks like in an actual photomicrograph. Now here's what's really cool. So let's just draw. Here's the syncytial trophoblast. Oops. Okay, so there's that. Okay, then the cytotrophoblast is going to be there. So this is all, those cells have all just kind of oozed together. And then let me get a different color. Here is the edge of the endometrium right here. And so this sucker is almost implanted. And so the endometrium would then be touching up here. And it comes down here and then right here. So see how it's not there, but it'll grow over. So, you know, in one more day after this picture, then this will have come all the way over to here and it'll be totally implanted in the uterus, in the endometrium. Okay, so I keep telling you about the hydatidiform mole. So here is what happens is that the fetal tissue, the placental, the trophoblastic tissue starts growing weird, and it can be cancerous and stuff, but it's not, it can just be benign. It doesn't necessarily have to be cancerous. And so I just want to show you this. Oops, not that. Share screen. So here is a hydatidiform mole is growth of an abnormal fertilized egg and overgrowth of tissue from the placenta. Women appear to be pregnant, but the uterus enlarges much more rapidly than a normal pregnancy. I don't know that mine really did, but so with mine, there was no, let's see, where does it talk about? There's no, oh, here we go. Abnormal fertilized egg develops into the mole rather than a fetus. And then there's no baby that's there. It says rarely a mole can develop when there is a living fetus. In such cases, the fetus typically dies and miscarriage often occurs. And so I'm going to show you a picture of what it looks like. What you do is, oh, and so here's the thing is ultrasonography blood test to measure human chorionic gonadotropin, which is produced early in pregnancy, and a biopsy are done. There was HCG being secreted by this structure, and so you know, you think that you're pregnant when you're actually not. You just have this tissue that's growing. It says 80% of them are not cancerous. And then women who have hydatidiform moles feel as if they're pregnant. Nausea, vomiting are common. Vaginal bleeding, I started to bleed, and that's when I thought, you know, that I was having a miscarriage. Parts of the mole deteriorate. Small amounts of tissue which resemble a bunch of grapes may pass through the vagina. They'll do a D&C. Dilation and curettage, which is when they scrape out your lining of your, well, suction out, but they clean out the lining of your uterus to get rid of any of those products of conception. Make sure that I'm still sharing this. Okay, so this is what's going to happen. We can have it if you get two sperm fertilizing an ovum at the same time, so that you get a 69 chromosome. So there may be some fetal tissue present, but you can't grow into a human with that. You've got way too many chromosomes, so it won't develop. This is what it looks like. kind of gross looking. So this is part where the placenta is. And so you can see all these little grape-like clusters, and that secretes lots of HCG so that you think that you're pregnant when actually there's nothing there. Okay, so so we've implanted, and so here's what's going to happen as far as that HCG is concerned. So we need to produce the HCG to tell the placenta, to tell the corpus luteum to keep making progesterone. So you can see ovulation, fertilization, there we are, 2 weeks. So week 0, this is really lame because nothing happens. This is where fertilization happens, as week 2. This is the first 2 weeks. So this is the first day of your last missed period. 2 weeks later, you got pregnant. And then 2 weeks later, you got implantation happened. And so now our HCG levels are going to significantly rise for the next, for the next 8 weeks. And then, so here about 8 weeks is where we're going to have our highest levels, and then they're going to gradually drop off. So by the time, and then all of this time, our progesterone levels are rising with this HCG that's being secreted. And the placenta is working on making its contribution to it. But right here, when we get to 14 weeks, okay, so we're now into the second trimester, that's where we don't need HCG anymore, because at this point where the estrogen and progesterone goes up, this is the placenta making the estrogen and progesterone, and then it will continue to make it for the remainder of the pregnancy. So I guess in my situation that my progesterone levels are like super way down here. I don't know why in the first case that my child died, why my child's heart did not continue to beat. I don't know, a lot of different things, but anyway, the idea was that let's put you on progesterone, let's keep you pregnant, let's hope that everything, maybe because I didn't have enough progesterone, that the baby wasn't getting enough nutrients from what placental tissue had established at that point. A lot of, I don't know, had no idea. Most of the time we don't know either. We just have a miscarriage and then we go on. So anyway, that's that. So this is that magical point right there, is when the placenta takes over, and then the corpus luteum by this point is not going to be doing anything anymore in the mom. We don't need it, so we don't need any more HCG. Now, just on a little side note, because I don't know how much we've talked about this before, why would people, because there was a thing about 6 or 7 years ago when people were taking HCG as a weight loss thing, and I thought, why in the world are men taking HCG? Because that's really lame, because men are never going to be pregnant. So I guess that men, I don't even know why. Would men have HCG receptors? Doesn't make sense to me. But what HCG does is when it comes out in the mom, besides letting the corpus luteum know that she's pregnant, the reason why you were losing weight is because we were liberating fat from adipose tissue to nourish the baby. So that's the idea. But people that were on HCG were also on 500 calorie diets. Duh. So 500 calorie diets what's making you lose weight, not the fact that you're taking HCG. So anyway, I don't know. It kind of came with a bang and went out with a whimper. And so anyway, that's that. Okay, so now let's make a placenta. So placentation is the process where both embryonic and maternal tissues will form this exchange organ to nourish the baby, which is, amazing. My daughter and I, because we're nerds and we talk about weird things, we were talking about the other day, mammal reproduction and how mammals, I guess oh, darn it. It's coming.
As you said it, it's not going to happen.
I know. Ugh. Okay. So mammals, birds at this point were well established. Well, birds have been around for a heck of a long time, because they're dinosaur remnants. And so birds and their amniotic egg, and crocodiles and their amniotic eggs. And then we had we had mammals, And you know what? I haven't really thought about those early mammals, you know, that made it through that Cretaceous Tertiary asteroid hitting the Earth thing and extinction. And then what was their reproduction like? Were they egg layers like the monotremes? So like the platypus and the echidna? And those are just the holdovers, and then we went into marsupials. I don't know. But anyway, so Missy and I were talking about how mammals, if the platypi were an offshoot, so eggs were some weird holdover, you know, but then we went straight to marsupials. So you have a little pouch, you know, and your baby, it doesn't really form a placenta embryonically. It grows, and I don't even know really what it does in the uterus of a kangaroo till it gets to that little embryonic stage. And if you've ever watched, oh my gosh, if you've ever watched an embryonic kangaroo make it into the pouch, its little back legs really aren't even developed. It just crawls up the mom. So it comes out of her vagina and it just crawls up her abdomen, and then it finds the pouch. And, you know, they look like those little pinky rats that have no hair and no eye, you know, just their eyes are closed and everything. And then it falls in the placenta, and then it finds a teat, and then it just latches onto that teat, and then it just drinks milk for the rest of its fetal development. So that's crazy. But then we have the placental mammals, which was, you know, really what we do, because that's the most successful way of caring for your offspring, is to build that exchange organ so that the mom can contribute nutrients to the baby and carry away the waste. And then the baby has the opportunity to absorb those nutrients and then secrete the wastes back to the mom. So it's such an amazing organ, the placenta is. So, sorry, I kind of got off on a tangent, but so the chorion from the cytotrophoblast will become the chorionic villi that we saw in lab. New vessels form from that, become the umbilical artery and vein. So remember, there are 2 umbilical arteries and 1 umbilical vein. Maternal and fetal blood are separated by the membranes of the villi. So the syncytial trophoblast, and we'll look at a picture of it in just a minute. So always remember that maternal and fetal blood do not come in contact with each other. We just have diffusion across the membranes from the cytotrophoblast. So there's no mixing of the unique cells within the baby, because what if it did? If you took the cells from the baby and they got into the bloodstream of the mother, what would she do immediately? Mount an immune response against it, right? And it would be a specific immune response. And so she would produce antibodies against that baby while the baby was growing inside of her. So that's why we can't have maternal and fetal blood mixing at all, ever. Okay, and then the chorion is the portion of the trophoblast forms the placenta and makes HCG. Okay, all right, so here's what I was talking about, the membranes here. So this yellow part, or this orange part, let me color something that's going to show up really good. We'll do this color green. All right, so this membrane right here that's going to go around the chorionic villi and then out into here and then around like this and then back around. So you get where I'm going with this. Okay, this is the syncytial trophoblast right here. So that is the outer edge of the placenta. And then what the chorion did was make the chorionic villi into, I don't think you're going to see that. We'll do it in purple. Okay, make that into the syncytial trophoblast and then develop into capillaries inside of there. Okay, so all of this, right, so this is all chorionic villi. So each one of these little leaves is a chorionic villus with the capillaries inside. Okay. yeah, so that's all I need to talk about that. Okay, so let's go on to the membranes that we have. Let's actually look at a picture of it first. And then we'll go back to what they are. So we've got 3 fetal membranes. We have, no, we'll still do it in purple. So we have this one out here. This purple one you can't see anymore is the amnion. We'll just put amniotic cavity. Okay, but there's the amnion, is this blue layer right here, and that one up there, and then this one up here. So this is membranes when like your water breaks, that's the membrane that ruptures to let the amniotic fluid out. Or if you have your membranes stripped, ugh, that's the worst thing ever. I don't ever wish that on anybody. I hated it when I was having mine stripped. So that's the amnion. Then we have the yolk sac down here. So that's, oops, well, down here, yolk sac, okay. All right, and then that's gonna come from the bottom of my bilayer discs. Here's the yolk sac right here. And then here is the allantois, and we'll talk about that hooking into part of your bladder here in a minute. All right, so the amnion, located under the cytotrophoblast, eventually becomes filled with fluid, or eventually surrounds the embryo with the cord in the middle. I'll show you that. Why do we have that? For buoyancy, temperature regulation, and movement. Actually, we can go back here. So here you can see this whole blue, that's now the amnion. So when we're stripping your membranes right here, that's what we're trying to strip, is through this, so that this fluid will come out, which tends to be a precursor for delivery. And so now this baby is floating within the amniotic cavity, and you need it to float. You need to have enough of that amniotic fluid. And as well, when you don't have amniotic fluid, then you have a lot of other issues. The baby won't develop as big. If you have oligohydramnios, the babies just don't achieve as big a size as if they had regular amounts of amniotic fluid. Temperature regulation, movement. Okay, yolk sac below the embryonic discs forms part of the gut, which we'll see in probably on Friday more than today. Produces the earliest blood vessels and blood cells, and the cells that are going to become the gonads. Will become the spermatogonia and the oogonia that are going to go through meiosis for reproduction. So the germ cells, the ones that meiotically divide, go through meiosis. And then the allantois connects the embryo to the placenta through the umbilical cord. It'll actually be a part of the umbilical cord. Becomes part of the urinary bladder, not, I should be put part of the urinary bladder. So in fact, I'll fix that right now. Because it doesn't actually become the bladder itself. That comes from endoderm, from the cloaca. Oops. Okay. Comes part of the, all right. You know, it's funny how obviously in my embryonic class, embryology class, whatever that is, I've learned a lot more than I ever knew before. All right, so here just shows the different stages that the blastocyst into an embryo. So now when we're fully engulfed, now we're an embryo. So here is the syncytotrophoblast, syncytotrophoblast that's going to become the membranes of the placenta. Here's the cytotrophoblast that's going to become the chorion. It's going to give rise to the chorionic villi, which are so these are the chorionic villi again, that are going to form the blood vessels, the capillaries. And then around here will be the umbilical artery and vein that will lead into the umbilicus, into the umbilical cord. Here is at 4.5 weeks, we've got this chorion all the way around the developing embryo. Here the yolk sac is degenerating, but you can see, oh yeah, it's still in part of the umbilical cord there. And now here we can see in this 7-week-old embryo, here is the amniotic sac. Here's the amnion with the fluid in here. Here's the very degenerated yolk sac because we don't need it anymore. And then the umbilical cord, we can see the umbilical vein inside of there. Here is the here's the placenta. Okay, so this would have been in a, that's a pretty intact miscarriage. So I'm not sure how they got that, but because that amnion is still attached. Okay, so now the embryo has to go through gastrulation. So gastrulation is when we produce the 3 germ layers that will then develop into the rest of the embryo. So let's go back to here, to this picture. So at 9 days, we're still the bilaminar disc, or the bilayered embryonic disc. So we have the epiblast and the hypoblast are the 2 layers that we have right here. Then by day 16, once we've implanted, what actually determines that you are an embryo is when you are this trilaminar disc. So this 3-layered disc of ectoderm, mesoderm, and endoderm. Then we are now into the embryo stage. Okay, so by the 3rd week, so by the start of the 3rd week, you know, so 16 days. Oh, sorry, my eye's really bothering me today. So by the start of the 3rd week, the 2-layered embryonic disc has become a 3-layered embryo. We have a primitive streak. I'll show you a picture of that. What that does is orient the embryo to a cephalic end and a caudal end. So you have a head end and a tail end. And then cell migrations are going to occur from the the ectoderm down through the primitive streak into the in between the 2 layers to form that middle layer of mesoderm in there. All right, so here is your embryo. So if we just cut off the amniotic sac part and the yolk sac part, and we just have our 2-layered disc. So here's our epiblast, here's our hypoblast right here. Here's our primitive streak. This will be our head end, this will be our tail end. This is the anus would develop down here. This is the pharynx, the opening of the pharynx. Because humans are deuterostomes, chordates are deuterostomes, that we actually form an anus first, and then it grows up into our mouth. And so protostomes actually form the other, right? Protostomes and deuterostomes, yeah. So like sea urchins and things less complex than sea urchins will form a mouth first that will then grow to their anus. So like we're going to take our little embryo and we're going to turn it sideways. We're going to look at it straight on. So here's the head end, here's the tail end. And so this is, of course, this is left, this is right, because we're looking down on top of it. So now we're going to section through this primitive streak right here, so this little gap. And here's what's going to happen to the epiblast cells. They're going to migrate through that primitive streak and then connect and fill in between the epiblast and hypoblast. And then it will become ectoderm. So by day 16, those migrating cells will have differentiated enough to now become mesoderm. And then we still have that endoderm, the definitive endoderm that we started with as the hypoblast. All right, so what do these layers become? So the endoderm, I'll show you a cool picture of that. The endoderm gives rise to epithelial linings of digestive, respiratory, urogenital systems and the associated glands. We'll look at everything that's there. The mesoderm is going to give rise to the skeleton, muscle, kidneys, reproductive organs, except the vagina, because that's going to come from the cloaca, from the endoderm. Circulatory system, dermis, dentin of your teeth, always the middle stuff. So your heart is going to come from mesoderm. Yeah, that pretty much gives you the whole generalized list. And then ectoderm is going to give rise to the epidermis of the skin, the nervous tissue, all of the nervous tissue, sense organs, enamel of the teeth, lens of the eye. Your eyeball formation embryonically is actually pretty cool, because you have surface ectoderm, which is, you know, the covering over where your eyeball is going to be, and then you have neural ectoderm, which is the optic nerve that's going to come out of your brain, and it's going to head out to the eye region, and then you're going to form a little cup of neural ectoderm that's going to make like the retina and the choroid, and then that's going to come in and go over where the sclera and the choroid and the lens of the eye are going to be, and so that surface ectoderm comes around and sits in the little cup of the neural ectoderm, And oh my gosh, eyes are so, so amazing. They're just, I can't even tell you, friends. Just everything I learned in that class was so great. All right, so this is stuff that you already know. So we learned this. Here is the back of you. So you've got this section right here in the very center, the very sagittal portion of your back, mid-sagittal portion, that this is going to develop into your nervous system, so your central nervous system. And then the nodal cord, if you guys have been in Mr. Henson's class, and you'll know what makes a chordate. A chordate, you have pharyngeal gill slits. So embryonically, you have pharyngeal gill slits, and you have a nodal cord, dorsal hollow nerve cord, and a nodal cord. And I think those are the three that mean that you're a chordate. And so this nodal cord develops really super early, and it's going to end up persisting as the nucleus propulsus of your intervertebral discs. But then the somites, and I'll show you somites in a minute, are going to form the bones. So the somites of mesoderm are going to form the bones around, and then the outer portion, the connective tissue that goes out and around the outside part of your intervertebral disc. And so nodal cord used to be a good flexible disc that you had running along your back, but you turned it into a bunch of other stuff. I mean, actual vertebrae as you developed. So that's what the nodal cord is. If we look at a cross section of you, so this line right here is this picture right here. So we've got the ectoderm on the outside, and then we have this mesoderm or mesoderm that's going to grow out. We have multiple kinds: lateral mesoderm, intermediate mesoderm, lateral plate mesoderm, splanchnic, somatic mesoderm. So many different kinds of things that it's going to become. But so that's here at 17 days. 19 days, we're going to start to fold in. So you can see that neural plate is folding over. So here the sides are starting to come together. And we have this little edge of the neural groove called the neural crest that's going to descend down here. And I'll tell you what the neural crest cells form, among other things. So here's your neural groove that's eventually going to form your spinal cord, that neural tube. Okay, so then if we go to 20 days, here those neural crest cells have migrated inward underneath the surface ectoderm. Here's that same neural plate that's going to fold over to become this neural tube right here. We're going to get these, the mesoderm is going to differentiate into somites and intermediate mesoderm. And then the lateral plate mesoderm, splanchnic, and somatic are going to give rise to the wall of your thoracic cavity. And let's see, so that's what splanchnic's going to, what does the somatic, I can't remember now. Intermediate mesoderm is what's going to give rise to your kidneys and your gonads. And then the somites are going to give rise to your bones and muscles and tendons and all that kind of stuff that's out in your limbs and everything. So you're going to get these 33, I think you get 33 somites that are then obviously going to form your vertebrae. And then they're also going to form, that are going to go out into your limb buds and form those. These neural crest cells that were right here have now gotten bigger. One of the things is that as these somites form right here, they're going to take neural crest cell tissue with it, and then that's what's going to give you the dermatomes. So all of the nerves that are in all of the portions of your skin, so all of your spinal nerves, so C1 through the coccygeal nerves will all come from here and go with the somites out to the various parts of your body. Neural crest cells will also go out and make the ganglia of the autonomic nervous system. They'll also produce the adrenal medulla. So lots of cool things that neural crest cells make. Lots of things neural crest cells make. Okay, now how are you going to get a gut? So I don't have a gut yet. I have a neural tube right here, but now see how I'm still open. So let's go back here to this picture. I'm still just a flat little embryo like this. How come I don't have a cavity? I don't have a body cavity yet, and I don't have a gut tube or anything like that. Well, that's what we're going to form now. So here are our 3 layers. So there's our ectoderm, our mesoderm, our endoderm. We're going to get a dip in here. So we're going to get a dip along our back that's going to make our spinal cord. And we got the somites that are going to give rise to everything, all of the middle stuff, muscles and bones and all that kind of stuff. And then we're going to fold over and we're going to seal off this tube right here. So here I have all of this endoderm, all of this yellow that's going to pinch together, and then it's going to descend from the mesoderm, and it's going to be suspended within your abdominal cavity and your thoracic cavity, and I'll show you how that happens. So we're going to get 3 sections to our gut. We're going to have a foregut and a midgut and a hindgut, and this is eventually what your gut's going to look like. So in your foregut, you're going to get your nasal cavity, your pharynx, and your throat. Then we're going to get little branches that come off of that. So we're going to get the thyroid gland that comes off of that. We're going to get parathyroid glands and thymus that branch off of that. We're going to get off of this initial gut tube. So this is all epithelium that this yellow thing is made out of. We're also going to branch off of the front in the larynx region, and we're going to build a trachea that's then going to come down here, and then we're going to build lungs off of that trachea. And then the esophagus is going to continue. Then we're going to get to midgut once we pass through the diaphragm. So like the diaphragm is going to come from mesoderm, all that stuff it's beautiful. And then we're going to get a stomach, and then off of our gut tube. So right here, our liver will sprout. Actually, it sprouts right here. By our pancreas will form 2 parts. So our head and the tail of the pancreas will form, and then they'll come together to produce 1 organ. And then remember that the liver and the pancreas, their ductwork all comes into the duodenum in the same place. That's because they grew off of the same area. So here's our liver that's going to grow off of our gut tube. And then our gut tube's going to come around, and it's going to be our small intestine right here, and then it's going to loop around, and it's going to be our large intestine down here. Now we're going to get to, and this is about where I'll end, we're going to get to this part right here. So this right here is going to be, at this stage, the cloaca. Okay, so you know a cloaca is a urogenital and digestive system opening. So you're going to have reproductive organs and urinary organs and digestive organs all dump into a cloaca. So birds have cloacas, reptiles have cloacas, and so that's why when a chicken lays an egg, sometimes it's got poop on it, because it all comes out the same hole. Well, we subdivided our holes. So females made 3, boys made 2. So here's what's going to happen is that mesoderm is going to come in here and split this. We're going to get a urogenital diaphragm thing, septum, actually urogenital septum first, that's then going to give us the bladder. Where's my bladder area pointer thingy? I don't have a bladder. Okay, so this is going to give rise to your urinary bladder, and then here is that allantois that's in your umbilical cord, and that's eventually going to become the urachus. And that's why I said if that doesn't seal over, sometimes babies can have urine that goes out their belly button. And then this will become the rectum down here. So this is the bladder up here, and this is the rectum down there. Okay, so there's how that forms, and that's all from endoderm. And then, so here is how your body cavity wall, so here is the yolk sac. That's that's where your yolk sac is, but our endoderm. So here's our endoderm is yellow. And so here is our gut tube. So what's going to happen is that we're going to seal off our connection to the yolk sac and right here. Then splanchnic mesoderm is going to become this visceral peritoneum and our visceral serous membranes. And then here's our parietal serous membranes from the somatic somatic mesoderm, the lateral plate. And so then these come together. So then you can see these things all unite and then detach from the yolk sac. and then here I have. So I have my body cavity. So I have the coelom or the peritoneal cavity, and then I have my gut tube, and then I have peritoneum. So like is going to go around all my organs, and then but because I get stuff forming outside here in my intermediate mesoderm, that's why the kidneys are not in the pelvic cavity, in the abdominal cavity. So you can see how the peritoneum, let me draw instead of just do it in purple. Okay, so see, here's my peritoneum, and my kidneys just poke into it, but they never get inside of it. Okay, so here, this is all parietal peritoneum right here. Well, visceral peritoneum, parietal peritoneum right here. Okay, so parietal serose, and then this is the visceral serose, and then they unite in the middle. Okay, but that's why the kidneys are retroperitoneal. A third of the diaphragm, or a third of the duodenum is retroperitoneal, and all of the pancreas is retroperitoneal. They're all poked back here on the other side of the peritoneum. Okay, so I guess that's, oh, here's the other thing. Okay, so this is, what else splanchnic mesoderm makes, now that I remember looking at this. So this is just your epithelium right here. So this is just simple columnar epithelium. But remember, you need smooth muscle for peristalsis. So that smooth muscle is going to, remember I said mesoderm gives rise to muscle. So here is that splanchnic mesoderm is going to form around your gut tube, and then it's going to form the smooth muscle that would be in, so the longitudinal layer and the circular layer of smooth muscle that goes around your gut tube, that comes from your splanchnic mesoderm. Okay, I think that's probably enough. Like I said, I could go on for days, but I'm not going to. So yeah, this is a good place to stop, because we want to have a baby on Friday, and we want to make them boys and girls. So we'll stop at this point. This just shows what happens to your embryo until it becomes a fetus. Okay, so here by 8 weeks, 9 weeks, everything is in place. Now we just need to make it big. And so at this point, when you become a fetus, everything's in place. We're just fine-tuning what it looks like. But we have laid out all of the plans of everything to become what it's supposed to be by the time we're at the end of our 8th week. So fertilization, 1 week conceptus, 2 week conceptus. Now we've implanted and we become that 3-layer disc. So we have red, blue, and yellow, and so we're an embryo, and then we're going to keep growing, and then we become a fetus, and then from here on out, it's just a matter of getting bigger. All right, so we'll stop right there, and that's all. So anybody have any questions? Oh, sorry. So Olivia said, what layer of the uterus does it implant in? And Trevor said the functionalis layer, and Olivia said thanks, because that's the right answer. So it goes into the functionalis layer, because that's the layer that sheds. So remember, we build that functionalis layer in hopes that a baby implants. And if the baby doesn't implant, we shed that whole functionalis layer and we make a brand new layer for the next month, for this next opportunity for a baby to implant. So we're always cleaning house in preparation for a new tenant to move in. Okay. All right, so I guess that's all. Sorry, I didn't even notice that in my chat. I was so excited about teaching you other stuff. So thanks, you guys, and always do this. I love that you guys help each other out when I'm just off in my own world and I'm not even aware of what you're doing. So keep it up. I love you guys. You are, okay, so I'm gonna cry, but I want you to know this, that you guys have really been a great cohesive group. And what a great group of people to be with during this really trying time. You should be fortunate to have the people that you have in your class, because they're the best. You guys are wonderful, and I love how much you help each other and care about each other. And if you haven't felt that so far this semester, then don't be afraid. Maybe reach out, because everybody's willing to help everybody else. And I love that so much. I love you guys. I'm going to miss you guys so much. And I just hope that everything is great and wonderful for your life in the future after all this crap is over with. And so I just, thanks for that. I don't know how to do those little, people were doing in a meeting yesterday, doing the little hand clap and the little thumbs up thing. But seriously, you guys are absolutely the best. And I just am going to miss you so much. So anyway, but I'll see you on Friday and I'll see you on Monday and Wednesday. And then, I figured out, did I tell you yesterday how I wasn't sure that we could have the final on the 7th because something was messed up? Well, I figured out and it's fine. So we'll have the final on the 7th, but I'll talk more about that on Friday when I get a little bit more put together and more about what I'm going to do about the final, because I still haven't exactly decided how I'm going to run it. I keep mulling over my options. It will be, you'll be where you are. You'll be at home or wherever you are. You don't come to campus or anything like that. But exactly what I'm going to do, I don't know, to ensure test security. That's still my issue. I know what I want to do. And I know that some of you are going to go, no, don't do that. But anyway, I'll pray about it. I'll decide what I'm going to do. I don't know. Yeah, that inspiration will come to me in one form or another. I'll meditate. I don't know. Okay, so I'm done rambling. You guys have a great day. And I won't see you tomorrow. I will see you on Friday. Okay, bye.